Welcome to Physiology Bites, everyone. We're doing something a little bit different today. We're going to do how muscle contraction occurs in Minecraft. So we're going to go from the very start to the very end. So let's go to the very beginning. When you decide you want to contract a muscle, whether it be because of a reflex action or you want to run away from something, okay, or you're just lifting the TV remote, whatever you're doing, it's because your central nervous system will tell a motor neuron to release a neurotransmitter called acetylcholine. And this acetylcholine is what we call excitatory, meaning it activates the thing which is after the neuron. So let's have a look what that looks like. We've got our big neuron here, okay? This is what we call the axon terminal, the end of a neuron, which releases a neurotransmitter. So let's pull the lever and release some acetylcholine. Okay, now in Minecraft world, we've used water to represent this, but you can see our acetylcholine only binds to certain points. Okay, it only binds to these specific receptor points. Let's have a closer look at them. So when we turn around, we can see that this is the area the acetylcholine has been received. There's specific receptors, the nicotinic acetylcholine receptors. Now in the body, in reality, this is a lock and key mechanism. So these receptors are in a certain configuration or shape that only acetylcholine activates them. And so when acetylcholine activates them, what they do is it allows sodium influx in this whole area here called the motor end plate, which is just a part of the muscle membrane, the sarcolemma, which receives acetylcholine. Now, when this acetylcholine causes sodium influx, it depolarizes the motor end plate, meaning the voltage becomes more positive. All right, and it becomes more positive because sodium is a positively charged ion, a cation. So when those cations start to come in to the muscle, the voltage goes from a negative voltage to a positive voltage, and this is the spread of an electrical signal that we call an action potential. This action potential will now spread from here in the motor end plate and then spread across the sarcolemma, which we have here in orange. And this is just the border of a muscle cell. And let's see what happens now when this action potential starts to spread in the sarcolemma to these things over here. These are voltage-gated sodium channels. All right, let's have a look at them. Okay, all of these are voltage-gated sodium channels. And we can see when they receive an action potential, because they're voltage-gated sodium channels, they're sensitive to changes in voltage, which means an action potential comes along and it causes the opening of these voltage-gated sodium channels. When these voltage-gated sodium channels open, it starts to depolarize that part of the sarcolemma. So what ends up happening is you get this propagation of an action potential. The initial depolarization, which starts at the motor end plate, causes cation influx into the muscle, which then spreads to the sarcolemma, which opens more sodium channels, which continues the action potential to be spread. And just like a circuit, which is what I've shown here, that action potential will spread down across the sarcolemma. It will just keep going, all right? But it will also go down into the T-tubule system. Now, this T-tubule system is a perfusion system which runs perpendicular to the muscle and where all the sarcomeres are, which is the element where all the contraction occurs. Now, because they run perpendicular, they allow for a lot of perfusion. So all that extracellular fluid, which is outside the muscle, has multiple access points to where the contraction needs to occur. So when we go down the T-tubule, we can see that what's happened is that action potential spreading down the T-tubule via the opening of voltage-gated sodium channels has caused the opening of calcium channels, specifically these channels here, the dihydropyridine, I've used abbreviations here, dihydropyridine and the ryanodine receptors. Okay, the dihydropyridine receptors act as little tripwires. They detect the change in voltage and they cause a configurative change to ryanodine receptors of the sarcoplasmic reticulum, and these ryanodine receptors open and release calcium. So calcium flows into our sarcomere. The reason it flows so readily into our sarcomere is because of concentration gradient. There's lots and lots of calcium in the sarcoplasmic reticulum, my apologies, but not much in the actual sarcomere. So therefore, when calcium is released, it will go into this area which has low calcium levels. So this is a diffusion gradient. We then, if we have a look at this sarcomere, let's see what this calcium will do. This is the action of calcium. Calcium will cause cross-bridging, all right? It will cause, I'll do it a few times so you can see it. It's a subtle action. Those pink blocks are the thick filament. The green blocks are the thin filament. And those red blocks are the myosin heads from the thick filament. So those myosin heads, the red block, are bound to our thin filaments in green. And then when we have contraction, it's a power stroke. So the myosin heads move the thin filament a small distance. You can see it's a small action, but it's there. This is a power stroke. All muscle contraction is a myosin head binding to a point on that thin filament. It's an actin binding site. It binds to that point on the thin filament. The power stroke occurs, as you see here. It then detaches, and then reattaches to a different point. And the great analogy of this is imagine you only have if you have one hand tied behind your back, but you had to pull a rope. You'd pull the rope and then grab it at a different point. This is what those myosin heads do with that thin filament. They grab it, pull, which we call power stroke, and then they reattach to a new point. And that's the reason your muscles, if you flex your biceps or anything like that, it's all happening because all these millions of sarcomeres, which is in your muscle, all do this action. The thin filaments sliding past the thick filaments because the myosin head pulls on that thin filament in this action. So this little action is muscle contraction, just repeated millions of times. All right. Thank you, everyone.